Hello, my name is Jenna. Today I'll be presenting the case report titled Genetic Exploration of Dravet Syndrome, 2 Case Report under Complex Genetics. I will first walk through what Dravet syndrome is genetically, then talk about the 2 patient cases and the clinical implications. So what is Dravet syndrome? It is characterized as severe infantile onset developmental and epileptic encephalopathy, or DEE. The article didn't specify the inheritance pattern, but during my research it came up as autosomal dominant and that most of the known cases are de novo. The onset of Dravet syndrome is within the first year of life. Dravet syndrome is caused by mutations in the SCN1A gene and is characterized by drug-resistant, intractable seizures, developmental delays, and comorbidities emerging over time, meaning that a person with Dravet syndrome will experience other medical conditions at the same time. Seizures of Dravet syndrome can be triggered by things like infection, hot environment, exhaust, sunlight, or exercise. In this specific case study, the 2 children were diagnosed with Dravet syndrome after getting seizures triggered by post-immunization fever, but I will talk more about this in the future slides. Early recognition of this syndrome is important to ensure the right treatment choices that won't cause more damage to the individual. The genetic background of Dravet syndrome. Dravet syndrome is caused by a mutation in the SCN1A gene that encodes for the NaV1.1 sodium channel that is needed for proper neuronal firing. A mutation in this gene causes the sodium channel to lose its function, leading to hyperexcitability and seizures. Most Dravet syndrome cases are caused by de novo SCN1A mutations. In this specific article, both children in the case study had pathogenic SCN1A variants. The aim of the case report. The authors in this case report describe 2 Indonesian Javanese children with Dravet syndrome, focusing on their clinical features and genetic findings. And at the end, they highlighted the challenges of diagnosing genetic disorders like Dravet syndrome in low-resource settings and the importance of next-generation sequencing. So starting with the first patient in case 1 involved a 11-year-old boy with uncontrollable seizures. He first developed these seizures during infancy at 3 months old after receiving a tetanus immunization that was accompanied by a fever. He had been born to healthy parents and had a younger sister with normal development. He had experienced developmental delay and was not able to perform certain motor activities. So this is the genetic findings for the 11-year-old boy in case 1. The little boy had an EEG taken as well as an MRI, and it was concluded that both tests were abnormal. The EEG showed epileptiform abnormality, and the MRI showed a bilateral frontal enlargement, empalomagria, which is a birth defect in the brain, leading to the symptoms he was experiencing. And lastly, they found a cyst in the right temporal lobe. After these results, he was referred for genetic testing. After whole exome sequencing, or WES, showed a copy number variant, they did whole genome sequencing, WGS, on the boy and found a large deletion of about 554 kilobases on chromosome 2q24.3. This deletion removed parts from 3 genes, TTC21B, SCN1A, the key gene linked to Dravet syndrome, and SCN9A. After getting these results, they looked at genomic population databases and found this deletion was not in the databases. They did, however, know that SCN1A is known to cause disease when one copy is missing, and other patients with similar deletions in this region had Dravet syndrome. The variant was then classified as pathogenic and confirmed the boy's diagnosis as Dravet syndrome. This is case 2, patient 2, and it's of a 1-year-old girl experiencing myoclonic seizure at 3 months old after receiving a tetanus immunization as well. After experiencing the seizure, the girl could no longer hold neither her head up or be able to hold a toy. This is the genetic findings of the 1-year-old girl. A CT of her head showed no major abnormality, but the EEG showed abnormal epileptiform. The girl was then referred to genetic testing. Whole exome sequencing found a pathogenic mutation in the SCN1A gene that is in a splice site before exon 24 of the SCN1A gene. They then used a computer prediction tool called Splice AI to predict what this mutation can cause. The tool showed that the mutation would likely do 3 things. First, it would remove 22 base pairs at the end of exon 24, then cause a frame shift starting at the amino acid glycine 1342, and finally lead to a non-functional SCN1A gene. The child was then diagnosed with Dravet syndrome. Sanger sequencing of the mother came back negative, and the father wasn't tested, so the inheritance pattern is either de novo or inherited from the father. I will go over the Sanger sequence results in the next slide. Next, I want to talk about before and after genetic sequencing. So in both cases, before genetic sequencing was done, both patients were being given meds that were not showing significant improvement in their conditions. In case 1, the 11-year-old boy had been given valproic acid, phenobarbital, and oxcarbazepine, along with therapy. After the genetic sequencing and his diagnosis of Dravet syndrome, the oxcarbazepine was switched to levetiracetam, and he became seizure-free. In case 2, the 1-year-old girl was being given clonazepam, valproic acid, and phenytoin that were not helping her condition, and after the genetic sequencing, they told her to stop the phenytoin and adjusted the amounts of the valproic acid and clonazepam, and her seizures then improved significantly. So before the genetic testing, these kids were being given medications that block the sodium channels and worsen seizures in Dravet syndrome. This shows the importance of genetic testing, and this example demonstrates precision medicine like we talked about during class, because it uses the patient's genetic results to guide treatment and avoid medications that worsen SCN1A related disease. Now I wanted to highlight the important key points from the discussion section. First, both children had mutations in the SCN1A gene, which is the main gene associated with Dravet syndrome. The clinical features and genetic findings matched what had been reported globally that SCN1A is the main player for the disease. Second is the authors emphasized the importance of early genetic testing and how that changes treatment. Before genetic diagnosis, both children were being given sodium channel blocking meds, which made the epilepsy worse. Once the genetic results were known, the treatment was corrected and their seizures improved. The third point is why whole exome sequencing and whole genome sequencing are important. Whole exome sequencing detected a splice site mutation in one child, while whole genome sequencing was needed to find the large deletion. This shows the need for different genetic tools to find different types of variants. The fourth point is that these cases add valuable information from an underrepresented population. There are very few, if not no, genetic reports of Dravet syndrome in Indonesia. So this helped expand the knowledge of SCN1A mutations in areas like Indonesia. And lastly, the authors connected these cases to precision medicine and how genetic information can guide therapy, avoid harmful meds, and tailor the right treatment to the individual. Finally, the key takeaways. To summarize, Dravet syndrome is a severe developmental and epileptic condition caused primarily by SCN1A variants and mutations. In both cases in this report, had pathogenic variants confirmed by next-generation sequencing. Early identification and next-gen sequencing is essential for the management of not only Dravet syndrome, but all other diseases and disorders. This report contributes valuable data from an underrepresented population that will help many other underrepresented communities.